Oh, I don't have to let people into this one.
Prof. Diaz, just so you know, it doesn't look like I can admit people myself. So,
Yeah, I don't think I put that I needed to admit people.
okay,
One I think
perfect.
I forgot. Yeah.
Um, do you mind if I make one quick announcement before you get started?
Oh, of course not.
Um,
Let
okay,
me turn this off.
I just got we both Prof. Diaz and I have been getting a couple questions about quiz 3. I just want to remind you guys there was a disclaimer at the very top of the quiz. We've dealt with this a couple times now, and each time we're sort of refining how those quizzes work, both so that the grading works well for you guys and for us. But please disregard your score until we've, and whether your question is right or wrong, your answer is right or wrong, until we've actually manually graded them, right? Because Moodle really has no idea what it's doing, except for like the multiple choice matching type ones. And because of the way it sort of allows us to grade and screws things up, I've reconfigured the question so the point allocation that you'll see isn't how it'll work when all the dust has settled, okay? So don't stress. And then finally, remember, recapture, recapture, recapture. Like if you're looking at, even after that, you look at your score and you're like, oh man, okay, look, it's just that's one step in the journey across a semester. You're gonna recapture, and you're gonna go into exam 1, and then you're gonna get that grade back, you're gonna recapture that grade. So, right, one step at a time, deep breaths, all right? And then I'm out of your hair.
All right. Sweet. All right. So got through that. So today we're going to be talking about plant nutrition and water transport. We're going to be continuing kind of our discussion, looking at how different chemicals that are important to plants as well as animals get into what we're going to talk about later as the greater food web and the ecosystem as a whole. And so, as we know, plants are primary producers. They are creating sugar and they're creating essentially all of the energy that's going to be used by organisms above them. So understanding how plants survive and kind of plant concerns in their environments will kind of give us a better picture of ecosystems as a whole. And so first thing I'm going to talk about is plant concerns, or this idea of, what kind of issues are plants trying to face? What kind of things are, actually, sorry, let me make this so you don't have to see the chat real quick. Cool. What kind of things they're evolving to kind of overcome and adapt. And so, for instance, we're talking about plants in an ecosystem. So plants need to grow tall. They need to avoid other plants. Plants aren't actively attacking or hunting for the most part other organisms. And so a lot of their interactions are kind of passive, passive aggressive, nudging against another plant and trying not to be shaded. Because if you think about it, every time a plant is being shaded by another, whether or not it's getting a lot of sunlight or it's getting a little bit coming through the canopy, all light that is not gathered is wasted energy. These plants are trying to produce energy. They are losing water through their stoma. And so all light that's not gathered during the day is wasted potential to grow. And as we said last class, plants are trying to grow constantly. They can, if they're given enough nutrients and enough sunlight, they can become, infinitely longer than when they were born. And so the problem is as these plants grow, they need to undergo both secondary and primary growth. So for instance, tall plants, not only do they need to grow taller and create more leaves to sustain themselves, but they also have to thicken those stems, right? And as those stems get thicker, they require more water, they require more nutrients. And so, an example of something that doesn't need this, though, instead of plants, are vines. Vines are plants which rely on others for support. Like tomato vines or ivy, they cling onto buildings, they cling onto other plants to hold themselves up. And so they don't need that secondary growth as much. And so they've kind of evolved away from that. But those plants that they're crawling on need to undergo that. And so that's going to be energy that they have to put forth. And so this is something that as you go in biology, you're going to see a lot of, which is the idea that all of evolution, all adaptations that animals have to their environment to try to survive are trade-offs. Nothing is ever going to be 100% efficient or useful. So, you want to grow tall and you want to become thick to outcompete the other plants in your area, right? But the problem is that if you get too big, you won't be able to feed yourself. If you're not collecting enough sunlight. So for instance, plants in winter, they lose all their leaves. Why is that? It's because they have less ability to collect sunlight. And so every day that they're sitting out there in the cold without an appropriate amount of sunlight, losing water through their leaves, they're going to be having a net negative effect, and they're actually going to be getting weaker. And so if they try to grow during that time, it's going to be harder. And so one thing that plants try to do that's really cool is they actually try not to overlap their leaves, because as I said, every stem and leaf is an investment. And so you don't want to be shaded by other plants, but you also don't want to be shaded by your own leaves. So if you look at, say, these two different plants here on the right, you have what is known as a 40% plant area. So you have these leaves. If you say make a circle around them, that's like all of the sunlight that is coming down onto them. All of this area that's not covered is wasted potential in order to gather light. And so when plants start to grow, instead of putting their leaves exactly underneath one another, they grow in alternating nodes. And so it gives the plant an ability to say, these leaves are at the bottom base of the plant versus these at the top. And so this ability allows the plant to now cover 80% of the light area. And so that's going to be way more efficient. They're going to be able to gather more resources and maybe be able to grow better. And so they're trying to maximize resource absorption. And this goes for both leaves as well as roots. If you remember when I talked about yesterday, roots will actually grow in ways so that they try not to intercept areas of low nutrients. So for instance, if a root feels that there's not enough nitrogen in the soil, it'll grow straight through that area instead of trying to adapt and say, waste time creating more roots or leaves. And so plants must place their leaves so they don't overlap. Ooh. So are water lilies highly efficient? So what do you mean by water lilies being highly efficient? So we have a question about that. Do you mean in terms of like the light they're affecting, or because water lilies, if you're trying to think of the ones that are growing out of the water and floating on the top of the surface, they have a lot of different issues. One thing we'll talk about is osmotic pressure and how lilies or organisms that are in water actually have to make it a point to not drown. So one thing we'll talk about with plants is that, not enough water is a problem, but too much water can also make them be sick. So water lilies would probably be decently efficient in terms of light gathering because they're floating on the top and there's not much there. But they're also, not creating these lower levels of leaves, right? Because that would just be completely wasted underneath the surface of the water. And so in some ways they're efficient, but in other ways they have it much harder than other plants. And so that leads to what is known as this phenomenon as shy trees. So I don't know if you've ever gone in the forest and looked up, but it'll often look like this. And if you notice, all of these trees, none of them are touching, which is why they call them shy trees. all of these trees, not only are they growing in a way so that they maximize the light that is only hitting them, but they're trying not to overlap with other plants. They're trying not to share that light resource, because again, growing underneath another plant is just wasted resources and not going to be nearly as efficient as they would like it to be. And so I always thought this was really pretty. I like this idea. You could just see like this breaking. It always kind of looks like starlight to me. You can see like speckling and things, but you know, trees are trying very hard to, you know, maximize everything without having to move very much. Yeah, trees, no touchy. Exactly. Cool. So as I said, plants have a number of so a great question is, how do plants know where the leaves of other trees are? And so what we'll realize, something we'll talk about in a little bit is actually how plants respond to different stimuli in their environment. And so one of the things is, not only do plants have photoreceptors in order to absorb light, in order to create energy, but they also have different photoreceptors that will let them know when it's dark versus like light versus dark, when they're being shaded versus not. If you ever, you know, had a plant in your house and you had it in the sunlight, it'll grow in one direction. If you say put a shade or close your blinds, that plant will usually bend its body and will actually try to aim towards a different light source if it can find one. And so plants know whether or not they're getting direct sunlight or some type of shading. And so, yeah, plants can sense light, gravity, and all these other stimuli, and we'll talk about that. And so in rainforests where everything is very, very dense and that plants are all in one area, plants will overcome this by, again, like vines will try to grow up other plants, or plants are actually, you know, if you think about a forest, often there's very little low-lying foliage if there are big plants at the top. And so it's really about how much light is coming through that canopy. Cool. And so, yeah, as Professor Aiken is saying, is all of these are molecular systems. They're molecules which are interacting with sunlight in some way that'll lead to this creation. And so another plant concern I wanted to talk about really quickly was this idea of osmosis and osmoregularity. The idea that plants need to take in water, but they also need to make sure they don't lose water. And so, you know, osmosis is diffusion, right? It's passive. And so plants really like this because it requires no energy and makes all of their systems more efficient. But the fact that it's passive means that you can passively gain water or you could passively lose water. And so plants need to have underlying mechanisms to protect against that. For instance, we were talking about those dermal cells. They have that waxy coating on them to try to keep water trapped inside of them. And so, you know, just like everything else, diffusion of molecules, you know, osmosis is going from areas of high concentration to areas of low concentration. And so there are generally two things that can happen, at least at the most extremes, when plants are dealing with different solutions. And so one thing I would like you to be able to do is do these kind of pressure calculations, and I'll go over that in a second. And so there are two different types of pressures that we're talking about in systems like this. So we have this psi p, which is known as the partial pressure. And so this is pressure that we're applying to the system. So for instance, let's say you have a syringe of a fluid and you're trying to inject it into something. When you push on that syringe, you're going to be creating positive pressure. You're forcing something to move forward. When you're pulling and drawing up liquid, say with a pipette or something, that syringe, you're creating negative pressure. And so obviously if you put pressure, you can force water through something that it's not supposed to. You know, it's kind of like that active transport. You're using energy to force something to move when it normally wouldn't. And so in these examples, I'm going to show you that pressure is going to be zero. We're not going to be applying any pressure. We're going to look at only passive movement. This other sign that we're going to show here, where it shows an S, the psi S, that's the pressure of the solution. And what we mean by that is that as we were talking about, water isn't pure all the time. You often have solutes in them. So you have salts, you have proteins, and so those different gradients can affect how water is going to move. Because say you have too much, in this example here, we take a cell that has a set concentration. So it has a set amount of salt and protein in its system. It's what we call a flaccid cell, because that cell is not having enough, doesn't have, it's in perfect equilibrium. You don't have anything coming in or out. And so we have this measure of the pressure, which is just the salt solution around it, which is -7. If we move that into a solution that has a lot of sugar in it, that proportion of water is now going to be lower. And so that increased sugar is going to result in increased solute pressure. And so this is what a solution is, what we're going to call hypertonic, which means that it has too many solutes. And so since there's too much sugar on the outside, that sugar is going to diffuse into the cell, and that's going to cause the water itself to leave. And so the cell's going to then shrivel. And so this is what happens if plants don't have enough water. If they have too much salt in the ground, it will cause those cells to shrivel. So just like SpongeBob, when he goes into Sandy's house and there's too much dry air, all of the water is leaving his body, and that plant's going to start to shrivel up. Now, the way you're going to know that's going to happen is that, again, in this system, we're looking at the pressure of the solutes. You have this negative pressure that's being caused by your sugars. And so the initial pressure is -0.7, and the pressure of the solute is -0.9. Since the cellular pressure is greater than the environment, so -0.7 is greater than -9, you're going to have that water flowing out. Now, in the opposite condition, we have stuff that is called, let's say pure water, distilled water that has absolutely no solutes in it. That's going to be the exact opposite problem. And so this is going to be like when Squidward ate too many Krabby Patties. So what happens is, once again, we're looking at this pressure. In this case, because the water is pure and has no solutes, there's going to be zero pressure being forced from the solutes. And so that pressure is going to be 0. Because there's now a lower amount of pressure inside the cell, the exact opposite is going to happen. And so in this hypotonic system, the water is going to flow into the cell, and the cell's actually going to get thicker and thicker. And if this cell doesn't have any way of getting rid of that water, it's going to burst. It's going to explode. And so the way I like to think of this is, hypotonic. I think of hippo. I think of it's puffing up. It's going to be big and fat before that cell dies. And so what I would like you to be able to do, and we'll have an example of this on the homework, is just compare what the total pressure would be in a solution and a cell, and determine whether or not it's higher or lower, and whether or not that cell is going to be in a good versus bad situation in terms of where that water is going to go. Yeah, so as Professor Ikin is saying, this is talking about PSI, or the pressure, the energy pressure. And so remember that water is going to flow opposite of the solutes. Because if you think about it, and this is a good point that I didn't mention, the solutes are not going to be able to diffuse across the membrane. So they're not going to be able to move in order to rectify this gradient that's being created. Since the water is capable of going over the membrane, it's going to be the thing that is changing and transferring. And so that's why organisms that can actually adapt to different types of fresh versus salt water, for instance, like bull sharks, they actually have mechanisms of pumping that salt in or out of their cells so they can create that gradient themselves.
I'm not trying to derail you, Prof. Diaz. I'm just trying to make connections to Module 1. So if you want to just, ignore them, you can, and keep your, mojo flowing. That's fine. Either way.
Oh, no worry. I honestly, I think it's a really good point to make that connection. So thank you. And so plants can actually force this pressure within their cells. And so this is known as turgor pressure. So if you ever seen a plant that's wilting, that's because it doesn't have enough water in it. Plants are using water to pump nutrients as well as to create stiffness within their bodies. And so without that, the plant is incapable of moving. And so they rely on stability and stiffness for this. When plants have specialized cell that allows them to move water, and this is how plants move. This is how they respond to sunlight. They bend by alternating where pressure of water is in within their cells. And so this image here is a basic looking at where the different resources are gotten from in a plant and how they're transported. Sorry, so let's see. Oh, yeah. So the question was, why is sorry, I'll go back to here if that wasn't clear enough. So the question was, why is the PSI 0 for the final turgid cell? And so, yes, so this is broken up into two different parts. Sorry. So you have the pressure of this one environment. And so there's the pressure from the cell is 0. We're not putting any pressure on it. We're not applying anything. And then the pressure of the solution, which is based on the pressure caused by the different solutes, in this case is also 0. So we have this one environment, and then we're comparing that to the same calculation before the cell was put into it. And so we're then going to be comparing this type of pressure. And so as Professor Aiken said, yeah. And so when you add this pressure to this pressure, you create 0 through it. Sweet. So we're going to talk about this. So, plants are getting each of the nutrients from different areas. For instance, first you have your water and you have certain minerals that are being absorbed through the roots. And so those different nutrients are going to be pulsed through the roots by negative pressure. And I'll go over how this goes through the plant in a later slide. And then this is caused by leaves losing water, right? We were talking about the stoma. And we were talking about how plants actively lose water throughout their leaves. And this is actually something that is both positive and negative. And that pressure from water being lost from the leaves of the plant is actually what helps drive up the water through it. And so evaporation of the water creates a tension force. So if you remember, we have hydrogen bonds that are happening between water, and water is very cohesive. It wants to attach to itself. And so as water is evaporating from the top of the plant, you have the water that hasn't evaporated pulling on the water all the way down from the roots. And since all of these cells are connected, it's one tight cord. Think of it as a rope of water that is being created inside the plant. When you when you remove the top molecule of water and pull on it, you're then pulling on all the other water that's at the base of the plant. And so you can have water being passively pulled up to the top of the plant. In terms of other allocation, you have, sugars that are being created, right? We have these sugars being created by light and CO2 in the leaves. And so that sugar has to be transported downward against this water gradient. And so the plant has two different sets of veins and veins, essentially, that allow them to do this. And so those sugars are not passively brought down. They're pumped by positive pressure. So the plant doesn't need to do any work in order to pull up water into itself, but it does need to actively do work in order to pump sugars down its body. And so that's one thing I'd like you to think about as we're talking about these two different mechanisms, how they're evolved to specifically passively versus actively pump things. And so the two different vessels, those vascular cells that we were talking about, those vascular systems that are being created, as I mentioned before, are the xylem and phloem. And so the xylem is, as I said, is using negative pressure. It's using that passive force of water to go through it. And as I said, the venation is similar in our bodies to veins and arteries in terms of one going, one way and one going the other. And so the xylem transports water and minerals, as I said, from the roots to the shoots. And this is a one-way flow. This is going from the bottom all the way up to the top of the plant. And so one thing that's really important to note about these is that there's no end walls between these cells. And these are those cells that I was talking about, especially in like woody plants and trees, where those cells are dead. They're born, they create the scaffolding, and then they die and they leave this hollow tube. And so, yeah. And so this, if you think about it, these cells in the xylem need to be dead, because if they were alive, they would have cell walls between them. They would have cytoplasm. They would have all of these things in them already that's going to impede the flow of water. And so having these cells be dead allow them to have no resistance of the natural solutions and the natural processes that are undergoing in that plant. And so those walls are stiff and they're made of dead cells and they're dry. Once again, if you have excess water, you're not going to have this gradient. So if they're dry and dead, you're going to force that water to move in itself. And so the phylum, which is moving against this gradient, is going to be transporting all of those products of photosynthesis wherever they need to go. And so the phylum cells are not dead. They are living, active cells that are connected to one of them, connected to one another. And so the cool thing is that these cells actually have holes in their cell walls that connect them. And so these perforations allow water to flow through, and they allow space for the cell to actually pump things through. And so one thing I haven't mentioned by name, but plants are creating sugar, right? What is in this phylum then if it's creating water and sugar? It's creating sap. So when you have a maple tree and you're putting, you're tapping sap and you're putting delicious maple syrup on your pancakes, what you're doing is you're actually finding this phylum in that plant, you're stabbing into it, and you're letting this sugar solution leak out of it. And so not only do we do that, but other organisms do that. And so sugar is that solution. So sap is not only delicious for us, but it's important for the plant to be able to pump throughout its body. And so since this pressure on this phylum, it has to be created, those cells need to be alive. They need to be alive to be able to actively move those sugars to the lower parts of the plant where the plant would otherwise die. Those roots are not creating any sugar for themselves, so they're not going to be able to create enough energy without this process. And the phylum also allows a two-way flow because it is active transport. So if they need to move something somewhere that's not necessarily purely down or up, or maybe a leaf is not producing as much sugar because it's being shaded, that plant might need to pump sugar to that area in order to keep it alive for a little bit longer. And so it allows it to actively decide where it needs things to go. And so this passive pumping, or this active pumping that's occurring, we've seen before. We've talked about in many ways in other processes in the cell, right? And so plants, as we said before, are using these hydrogen ions in order to act as a co-transporter. And so not only are they doing that in order to create energy, create ATP, as shown here, they're actually also doing that in order to actively pump sucrose, or that sugar. And so you can think about it as these have so many useful, these proteins just have so many uses. Obviously, it's going to be a different type of protein that's going to pull in each of these different types of chemicals, right? But they're all underlying this same kind of co-transporter system. And so in this case, those hydrogen bonds are co-transporting sugar across the membrane and causing that bulk flow, and they're creating that positive pressure. And so this is caused, this long distance flow is caused by specialization of the cells. You have specialized cells, which are creating and pumping and creating that pressure, while you have other cells that are acting as that structure in itself. And so these proteins that are located on the surface, the ones I want you to know about specifically are these aquaporins. And so those are the ones that help transport water across cell membranes. And I'll go over how they do that in roots in a second. And so when plants are transferring things throughout their cells, it's not as simple as simply pumping through the cytoplasm. There's actually a number of different routes that minerals and or water can take depending on where it's being uptaken and kind of how what that chemical is made of. And so, for instance, you have this aplastic route, which is going through the cell walls. And so going through cell walls. So you have water and solutes that are dissolved in that water. They can move along the continuum of the cell walls. And so they're kind of transferring and going between these cells and going between this hydrophobic layer, but they're never actually entering the cell. They're riding along that membrane and that outside area. You then have a symplastic route, which is when you actually have solutes moving through the continuum of the center of that cell. And so those are things that actively are being pumped or can move through. The difference between if something can go through each of these roots is whether or not it can actually be absorbed into the cell. If it can get through that initial cell membrane, it can then go through the cellular membrane. If it's hydrophobic and incapable of getting into the cell, it's going to ride along that outside line. And then you have this transmembrane route, which is sometimes you have different molecules that may not necessarily be able to go through these portholes. They're going to be actively pumped over the cell membranes. Or they'll be going through kind of like that phylum, those channels where you have holes in them and you're allowing chemicals to dissolve through it. And so you have the aeroplast, which is just everything that's external to the plasma membrane of the living cells. And then you have this idea of the simoplasts, which are consistent with the entire mass of the cells. So if you think about it, we were talking about how all of these different types of tissues are all interconnected. You have all of the cytoplasm of the cells connected throughout it, and that allows them to transport things very well. And so this idea of transferring sugars is known as translocation. So you're changing the location of things. And so specifically, we're looking at sugar being pumped opposite the way of water. And so obviously we know that sugar is being created in what we call a sugar source. So that's the plant organ that makes sugar. For the most part, that'll be your leaves. You have these leaves acting as a source. You have sucrose and water in there. And so those plants are taking sucrose, mixing it with water, creating this sap, and then pumping it across their cell membranes into this phylum. You then have what is known as a sugar sink, or a plant organ that uses and or stores sugar. So plants, similar to our fats, they can store sugar within themselves for later if they need to use it. And so a sugar sink could be just a vesicle or a vessel that holds that sugar, or it could be something such as a root which actively needs to use that sugar. But again, it's being pumped. And so you have delicious sugar and sap that are being pumped throughout this thing. And so this source cell, by creating pressure and pushing this sugar out into this phylum, is actually creating pressure in itself. And so it's kind of like when you use, say, a turkey baster and you squeeze the end, that pressure is forcing it out the end. You're essentially doing that. You're forcing sugar and water back into this tube. There's already sugar and water in there. And so it's going to create pressure that's going to force it forward. And so depending on where these cells are opening, if they need to send something down or send something up, these cells will actively pump in one direction to kind of regulate and fix that problem. And so again, it's being actively pumped using this co-transporter of protons to create this positive pressure. And so when you have, say, down here, this is a picture of, say, your root cells pulling in material, you'll have these two different paths, right? You're going to have the roots pulling in water. And so water being able to get into the cells is going to be pumped through these materials, pumped through these cell walls, and go through this symplastic route. Water also might be able to be pumped through this cell membrane depending on whether or not it's carrying certain other molecules, such as sugars or something that might not be able to be pumped through these smaller areas. Cool. Do I have time for this? So, oh, sharing screen. Let's share this one. Cool. And so, one example I'd like to bring up real quickly about this type of growth. Remember, we were talking about vines. We were talking about plants that don't necessarily need to grow in their own right, at least secondary growth. But there's actually plants that are vampiric towards other plants. So, for instance, we have these vines that have fallen, these seeds that have fallen onto the ground. They've started to grow, and right now they're trying to get up to the top of the canopy, but they need nutrients, and they're not going to be able to produce their own sugars. So what they're doing is they're clinging onto a neighboring plant. And so in this case, you have each of these vines, which are this parasitic vine, climbing up the body of this plant. And so what it wants to do is it wants to find a way into that phylum in order to get that sap. So what they'll do is they will ride along these different thorns, and they will look for a weak spot or a spot they can actually feel if a plant has proper nutrition in it. So plants have ways, just like our cells talk to each other, where they can interact with the cell walls of other plants and just figure out, are there nutrients here? Are you wilting? Are you sending out chemicals that say that this area is really poor? But what this plant is doing here is it's wrapping around, it's finding an area of that high sap, that high sugar, and then it's stabbing into it and growing as part of that plant, and it can now absorb some of that sap. And so it's actually going to be taking all of the sugar from this plant. Similar to that strangler fig that we were talking about before, except instead of just using it for support, it's now growing and absorbing. And so this is swelling up because it's actually stealing all of that sugar and sap. And so this plant will not only do that, but it will make its way all the way up to the top of the canopy. And so it would be terrifying if plants moved this quickly in like real life. I'm happy that, this is a time lapse. But I don't know. I just really like vines. I think they're super interesting in the way that they activate. But they will, grow up and try to get up to the canopy as much as possible.
How did the seeds arrive here
Sorry, would you say how did the seeds arrive there?
Yeah, because if the plants is already being vampired.
Mm-hmm. Yeah, so we'll talk about this a little bit later when we talk about development of plants. But they have a number of different mechanisms for that. So once this plant gets to the top, it's most likely going to create a type of flower. And then it's going to use that flower to disperse its seeds. I'm not sure whether or not this plant can self-pollinate or if it requires another vine in order to do that. But what'll happen is it'll create those seeds and then it'll drop them on the forest floor. And the hope is that those seeds are close enough to another plant. So these plants are going to create oh yeah, so here it is. These are the flowers. And so these flowers are going to be pollinated by some type of, bee or something along those lines, and then it's going to shoot its seeds out onto the grounds. And again, the hope is that they're going to scatter from wind or another organism, and they'll be able to land next to something that is a healthy plant. They're going to distribute a lot of seeds, and not very many of them are going to survive. But if you see, this vine has made its way to the top, and now it's completely engulfing the plants around it. It is taking up this entire environment now. Yeah, cool. All right. I don't think you can see my PowerPoint screen again. Let's go back here. Cool. Yep, so that plant is undergoing primary growth, completely ignoring secondary growth for the most part, trying to mostly cling on to other plants for support. Cool. So when water is being absorbed by the roots, water is going to be undergoing multiple pathways, right? As we were saying, depending on what's in it, depending on whether or not there's dissolved minerals of, say, nitrogen or phosphorus or any of these important chemicals we'll go over later for the plant development, it will be able to move either absorbed through the root hairs or going along the different paths of this. And so water and minerals cannot be directly absorbed by the plant until they enter that xylem. Until they enter that vascular tissue, the plant doesn't have access to it. Again, it's as if you had to collect your blood and then put it into your vein from the environment. And so roots are typically especially sensitive to water. As I said before, those dermal cells on the outside of plants are often in organisms, especially plants, are often specialized in order to absorb or interact with a particular chemical. And so roots, especially sensitive to water transport. And so they will allow water to go through those membranes, through root hairs into their actual cell body. And then water can just be transferred from cell to cell until it gets to that xylem. The problem is that in order to protect itself, there's this Casparian strip, which is a waxy material that blocks and insulates that xylem and phylum. Once again, the plant doesn't want to lose water to its environment. If it's dry out, if it's a hot day, it doesn't want water flowing back out of its body into the ground. And so this waxy layer keeps that in. And so it acts as a semi-permeable membrane. And so anything that cannot go into a cell, such as this water, will not be able to pass through it unless it can interact with that membrane. So it's a checkpoint. Certain minerals will flow in through this apoplastic root. It will go through the cell walls, but once it gets to this Casparian strip, it's not going to be able to move forward unless it can interact with that membrane and be either actively transported or it needs to be actively transported. Sorry, it's not going to diffuse because it's so hydrophobic. And so you have passive movement through the cells, and then you have specific chemicals that can get through it and get into the center of the plant, which is known as that vascular cylinder, getting into that xylem. And so this area here, so you have the epidermis, which is the outside of that cell, that initial barrier. But because we have a secondary barrier, we have this endothermis. And so that Casparian strip is known as the endothermis, which further insulates the plant. And so talking about pressure, we have this idea of transpiration. So transpiration is the loss of water at the top of the plant through those stoma that cause the positive pressure to pull water up. And so this is actually something that I study in spiders and moths a lot, is the idea of passively moving water. How do you get water to move from a place without having to exert any effort? And so flow is caused by root pressure. So what happens is you have water molecules that are coming into contact with these root hairs. Root hairs are actually a single cell thick. They're extremely thin, so water is very easily able to diffuse into them. that water is then put into and pumped, not pumped, but moves into that xylem, those dead cells. And so you have hydrogen bonding, so you have this adhesion of the water to itself, but you also have this really cool idea of capillary pressure. And so with capillary pressure, sometimes capillary pressure is a cohesive force. So in this case, what happens is water is attracted to itself, right? But it's even more attracted to the walls of that that xylem. And so what happens is let me draw on this. So what happens is you have, say, this is the xylem, and then this is the water, right? If you've ever had, say, a beaker or a graduated cylinder, you've noticed a meniscus, right? Water will often cling to the sides of some type of tube that it's in. And so that's because of cohesive force. And so in the xylem, this exact same thing is happening. The water is attracted to the edges, and so it gets pulled a little up. What happens, though, is if that force is strong enough, not only is it going to pull up the sides, but the water is still attracted to itself. You still have those strong hydrogen bonds. And so the hydrogen bonds are going to move. Why won't you let me draw? The hydrogen bonds are going to move the water and pull it forward. And so it's essentially like I'm going to cling to the walls and grab, and then I'm going to pull my body up. But what happens is that once that water has become level and it's equal, oh, well, the water's still attracted to the walls. And so it's going to create that exact same situation where you have a meniscus, and then that's going to again pull the water up. And so this passive force of I'm attracted to the walls, I'm attracted to myself, attracted to the walls, can happen extremely quickly, especially when you already have a water gradient, because you're losing so much water at the top. That water's evaporating and pulling on this center mass even more. Yep, this is why water wicks. Like if you've ever given blood, they'll often test your blood iron level. If you think about it, generally what they do now is they prick you, right? And then they take this little plastic tube, they touch it to your hand, and that's all they have to do. That's because of this cohesive force, this capillary pressure that's happening in that tube to pull your blood without them having to do anything is exactly what's causing the water to move through plants in order to get to the top. And so it's pushing water up, and then it's expulsing water. This is gradation is just the idea that plants expel water in the morning in order to create a dry enough environment so that they can force that water out. And it's different than dew, but it looks the same. Essentially, you have too much water, you place it on the top of the plant, that'll dry out in the sun, and that'll start this process of creating this water difference. And so it's capillary pressure, as I said, and it's a weak force. It's a weak force in general, but because the xylem has all of these nooks and crannies, it's very dry, it's going to force it to move. And so you can move, you can have water move like 45 meters. So, 45 feet per hour of water moving up this plant. Oh, I don't like when that's there. Erase this. Cool. And so you have this transpiration. So it's 11:20. I have like two more slides and a video. So if you can't stay, that's okay, but I will put this all up on YouTube. And I realize if I talk enough during the YouTube videos, it won't delete them from the video when I put on YouTube later, so it's okay. But last thing I would like to cover is this loss of water through the leaves. And so we've discussed this a little bit, right? I just want to talk about the mechanisms that kind of underlie this, these opening and this closing of this stone. the stomata. And so, obviously plants are covered in this waxy layer. We went over that to limit water loss. So the stoma are actually where most of the water is lost. For an instance, in an acre of plants, about 400,000 gallons of water can be lost per day just through the stoma. And that's roughly 90% of all water that's lost through the plant is through these. And obviously we know they open and close in response to water and CO2. And once CO2 is depleted, they need that in order to undergo photosynthesis. They then open up. And so these guard cells, the way they work is they actually work through water pressure as well. What happens is these guard cells, when they are stiff and filled with water, when they are bulging with turgor pressure, that is because they are filled with waters, they're swelling, and then they're pushing from one another. The plant actually does this by using potassium. So by pumping potassium into these guard cells, it creates a really dry environment. And that dry environment pulls water into those cells and again uses that turgor pressure to stiffen them. And so these cells stiffen and they push up against each other and then they open. During night, once we need these to close, the plant starts to actively pump out that water and that potassium, leading to them closing. And so we'll talk about how again plants look at this light and how they're able to respond to it. But light stimulus creates this accumulation of K+. In response to light, knowing that it's most plants, can plants are different, knowing that light is out, they're able to respond to that, and those lights start triggering chemical reactions that force and pump the potassium across the ridge. And so one thing that's really interesting that I never actually really thought about is this actually is how plants create a circadian rhythm. And so plants can actually get jet lag. If you take plants out of their normal environment, if you flash lights at weird times, you can mess with their natural metabolic clock. And so this is part of that reason, right? They're used to having certain amount of hours of being able to pump potassium versus not, and feeling those lights. And we'll go over that a little bit more. And so as I said, this transpiration is based on cohesion. So again, similar to the xylem, the stoma are actively working with water. And so this is just a graphic of this capillary network that I showed you. This is the cells that are making up the cell walls. And so you have these cells here, and then you have water in between them. And so water evaporates, the water vapor that is leaving disappears and changes this type of pressure. And so then you have this water-air interface where the water goes back down. And so that's then going to force that water to pull forward once again. Again, you're evaporating, water's disappearing, the water is attracted to these areas inside the cell, so it wants to pull them forward. And so you're then creating that tension once again and pulling it upwards. And so that's that negative pressure and that tension. And so the last thing I would just like to show is a quick video that kind of sums up these processes and shows kind of what I like as a more active video, kind of showing how these different chemicals move through. Oh, no, that is the end.
Transportation in plants. At the end of this session, you will be able to know the function of xylem and phloem cells and explain transportation in plants. All plants need water. Plant roots absorb water and minerals from the soil. Water absorption is mainly carried out by root hairs. These narrow-walled hairs greatly increase the area over which water absorption can take place. Water and minerals enter the root by two pathways: apoplastic pathway and symplastic pathway. If water and minerals move from cell to cell through spaces between the cell walls, then it is known as apoplastic pathway. In symplastic pathway, the water and minerals move across the cytoplasm and finally reaches the xylem.
And so you could see whether or not the cell is able, or that chemical is able to get over that gradient and get into the cell, is whether or not it's capable of doing that.
Xylem vessels are long, narrow, hollow tubes containing no living material. The xylem transports water and soluble mineral nutrients from the roots to other parts of a plant. About 95% of the water absorbed is lost through the stomata present in the leaves. This process is known as transpiration. The number of stomata is more on the lower surface of leaves. If transpiration happens throughout the day, then there will be a great loss of water. This is controlled by the stomatal opening and closing mechanism. During the daytime, water enters into the guard cells along with the potassium ions. This brings about a change in the shape of the guard cells, and the stoma opens. During the night, the potassium ions and water molecules leave the guard cells. Thus, the turgidity decreases and the stoma closes. Stomata also act as main sites of gaseous exchange in plants.
So, yeah.
Leaves make use of water and the carbon dioxide for photosynthesis. Glucose formed during photosynthesis is transported to the other parts of the body through phloem cells.
Apparently, I've been mispronouncing phloem.
Phloem tissue consists of less specialized and nucleate parenchyma cells, sieve tube cells, and companion cells. Transport of food in phloem can be up or down the stem to reach all parts of the plant. Hence, the transportation in plants is the function of xylem and phloem cells.
Cool. All right. Cool. All right. So let's see. So that is the end of what I have for you. Obviously, you have any questions, I can stay here. But concept questions are at the end. There's a lot of them this way at this week. But a lot of them are kind of trying to get into bigger concepts, such as why plants, that things we'll use later, such as why plants may evolve different types of adaptations to their environment, can plants versus general photosynthetic plants and things like that. Cool. So does anyone have any questions about anything?
Have a good weekend.
Oh, take care. Yeah.
I have a question. If you could you go over the pressure stuff one more time?
Yeah, sure.
Because it was honestly what was confusing for me is not the concept, but the number add-ups.
Yeah, the number add-ups of that part.
There was on the slide as opposed to the book a difference, and I think it's the book's fault.
Oh, really?
Yeah.
Yeah. So this slide that this is like from an older version of the book. I don't know if they updated it. because I was having a hard time finding the exact image that was in the textbook. Let me see if there's a difference.
So I think the part that might be tripping up is at the bottom there, where it says 0 plus 0 minus 0.7 equals 0.
Yeah.
That should say 0.7 plus minus 0.7 equals 0. And that's what it says in your book.
Okay,
Okay.
that definitely makes more sense.
Yeah, that makes sense. Okay, this old.
I think Prof. Diaz got let down by the book because the way he explained it is, exactly right.
Yeah, what is this?
In the first one, right, it's just saying the pH of the solution is it's written as it's like there's solutes in the solution. That's why it's written as a negative pressure, because it's the pressure that's going to be like pulling stuff away from the plant cell, right?
Yep.
Mhm.
And then yeah, and then the second one's just going in. Okay, thank you.
Oh yeah. They changed it in the new book. That makes way more sense. Okay.
Okay, thanks.
Any other questions?
I have a question about the translocation slide. I was confused on, or I was just wondering if you could go over one more time how the positive pressure is created.
Oh, okay. So the positive pressure is created in the same way that we've kind of been creating pressure before, by using co-transporters. So we're using hydrogen gradients in order to pump that sugar out of this source cell and into this sleeve tube. And so think about it as if you have you have a tube full of water, or sorry, you have like a hose full of water. If you're trying to connect a secondary hose, you're going to increase that pressure that's happening in that initial one. And so by essentially squeezing this turkey baster up here through using the transport of these hydrogen atoms, you're creating pressure inside of here.
Okay, that makes sense. Thank you.
Okay. And again, these are living cells, so they can actively change that pressure at different parts along this tube in order to get it to go to whatever specific one they need to. Any other questions? Cool. All right, I'm gonna
Once, going twice.
place. All right, I will stop the recording and sold.